Okay, so we made it this far, okay? This is going to be Perio chapter 13, our last chapter to review before this test. I'm so proud of you guys for making it this far. So excited. We're going to make this A tomorrow. This has everything. We've been through everything that could possibly be on the test. Fingers crossed there. Last chapter, okay? This is Perio chapter 13, okay? There's a few here, so settle in. We are almost through this. Let's get it. So, first question, Star. He wants us to know this phrase. One human mouth is home. And I'm so proud of you guys, by the way, for making it this far if I didn't say that. One human mouth is home to more microorganisms that there are people on the planet. Basically, what that means is there's more microbes in the mouth than there are people on Earth. Okay? Next, what is the plural word for bacterium? Bacteria, and bacteria replicate quickly, okay? All right. What is the cell envelope and what does it consist of? Now, this is not starred. A cell envelope is just the skin of the microorganism, meaning the skin of the bacteria. The cell envelope is the skin of the bacteria. It consists of an inner cell membrane, the cell wall, and in gram-negative bacteria, they have an outer cell wall. So 3 layers of their skin, really, bacteria's skin, inner cell membrane, cell wall, outer cell membrane, okay? What are the 2 groups of staining? So the two groups are based on the structure of the cell envelope. So depending on their cell envelope, you'll know if they're gram-positive or gram-negative, because the gram-negative is the only bacteria that has that outer cell wall as well, okay? So gram-positive and gram-negative, and this has to do with gram staining, okay? So, well, of staining, gram-positive, gram-negative. Okay. There's no star here. Streptococci shape versus staphylococci shape. Streptococci is 3 or more in a bunch. Staphylococci is 3 or more linked in a chain. So streptococci, bunch, strep, throw is a bunch of pain. I don't know. Staphylo, think of like a staff, like a shepherd's staff. Is that what they're called? A staff is kind of like a long link chain. Like, it's not really a chain, but you know, it's a staff, so it's a line, it's a chain, okay? 3 or more linked in a chain. Staff is long, okay? It's a chain. Streptococci, 3 or more in a bunch. This one's starred. What is gram staining? It determines bacterial properties and pathogenicity. Yep. So basically, gram staining determines what this bacteria can do, and is it a pathogen or is it not? Is it harmful? Is it not? Gram staining lets us know what kind of bacterial it is. So what does it do, and does it cause harm, and how? We have to know what kind of envelope it has to know if it's negative or positive, or, you know, what kind of effects it'll have on us. So, gram positive, there's a star. This is why I reviewed a little bit about the cell envelope, because we're like, gram positive, you know, what is that? So I want to get some depth to it. So gram positive star is a thick single membrane. Okay, it's thick single membrane. Remember, because only the gram negative has that outer membrane. So the inner membrane and the cell wall, that's all this gram positive is going to have. And it's purple when stained. It's associated with periodontal health, so good health. So in the positive, think positive, we want good health. So gram positive is in good health, and it's stained purple. We're doing good. Our dental hygiene color is purple. So gram positive is good health, and it's going to be purple. That's us. We're good. We're doing great things. We are purple. That stands for dental hygiene. So a thick single membrane, okay? Thick single membrane in a gram positive, and it's all good. Periodontal health, good health, stains purple. We're purple. Okay, gram negative. Now let's think negative. There's a star. It's a double cell membranes. Okay, so double cell, we know it has the inner, the cell wall, and the outer. So there's 2 membranes there. So gram negative is double cell membranes. It stains red. It is bad. It is associated with periodontitis. So gram negative, think bad. It is bad. Periodontitis stains red. Red is bad. No good. Ed. XXX, stop the periodontitis. Oh my goodness, that's bad. Red is bad, okay? So purple's good. Purple's us, that's positive. Negative, bad, red, periodontitis, double membranes, okay? So 2 wrongs don't make a right, okay? 2 membranes, okay? 2 wrongs don't make a right. You're bad, you're red, you're periodontitis, you're gram negative, okay? 2 wrongs don't make a right. 2 membranes. We only have 1. 1 star for us. 5 stars, but anyways. Are bacteria social creatures? Star. Yes, they are social creatures. They always present on the skin and in the digestive traits or tracts and respiratory system of humans. They're everywhere. They're social. They live together in a complex microbial community similar to human communities, multicultural and well engineered. And we'll get into their engineering here in a little bit in this chapter. But the community is attached to living and non-living things. They are social creatures. They will talk to each other. They'll go all throughout your body. They're social. They intercommunicate. We'll get into that too. But they attach to living and non-living surfaces. That is very important. They'll attach to anything because we'll get into this too. But if they're free floating in the mouth, they are more susceptible to be rinsed out or flushed out with some antimicrobial rinse, right? So they need to secure themselves. They need to survive. It's survival of the fittest in there. These are little communities, microscopic communities. They are little living organisms, okay? They want to survive. So do we all. Fish, anything that's living, anything, birds, crocodiles, alligators for Lily. Everything wants to survive, okay? Same thing with bacteria. Even though we can't see them, they're still living organisms. They want to survive. So yes, they are social creatures, and we're going to get down in depth in this chapter of how that is so and what bacteria does, which is my favorite. So here we go. Aerobic bacteria, star. Aerobic, have you ever, I don't think that's going to be relevant. Aerobic bacteria, so they require oxygen to live. When I think of aerobics, I think of like water aerobics. So, water aerobics, they're in the water, but they, like if we're doing water aerobics, we got to come up and get some oxygen after a while, like we love the oxygen. We require oxygen, okay? We can be underneath and be aquatic and everything, but we have to come up for oxygen, okay? We require it, okay? Aerobic. We're doing our water aerobics, but we need to come up for that oxygen, okay? Aerobic is oxygen. They require oxygen to live, okay? That's us. We're aerobic, okay? Anaerobic, so anytime you put an in front of any word, and it's kind of like not, okay? So anaerobic bacteria means that they cannot live in the presence of oxygen. They cannot do it. They are anaerobic. They dive deep down. So this is cool. They go down, down, down into our sulcus as far as they can go. Those are the ones that bury down. They're harder to get rid of because they don't want the oxygen. I'm going to verify if this is true, but whenever you're flossing, there's probably multiple reasons for this, but whenever you're flossing and you take it out and you smell it and it smells kind of funky sometimes, so that's because you're pulling that anaerobic bacteria out. They don't like oxygen, and once they hit the oxygen, they release these gases and toxins, and like it's a funky smell. So I heard a hygienist say that one time. So when you're pulling that out, and you smell it, that's anaerobic bacteria. And probably like old food and stuff, but that's a fun fact, right? So anaerobic means not aerobic. They don't want the oxygen. They don't need it. They literally are fish. They cannot have the oxygen. They are down there. Okay, so they cannot live in the presence of oxygen. So what's cool is hydrogen peroxide releases oxygen that helps destroy the bacteria. So if you have those anaerobic bacteria that do not like oxygen, you put a little hydrogen peroxide there. It's going to make those oxidating, I don't know if I'm saying that right, but it's going to make oxygen bubbles, and that helps destroy them because they cannot live in the presence of it. They will die. Okay. So facultative anaerobic bacteria. So facultative, I don't know how to explain that, but Facultative anaerobic bacteria, there's a star, can live with or without oxygen. They thrive in either environment. So facultative, they can do a little bit of anything. You know, faculty. So facultative, faculty, substitute teacher. She could do a little anything. She'll pop in here, pop in there, you know, facultative. She can live with or without oxygen. Well, maybe not her, but you know, the bacteria, with or without oxygen. Pretty easy. How does hydrogen peroxide affect bacteria? There's a star here, and we just went over it. So, how does hydrogen peroxide affect bacteria? Hydrogen peroxide releases oxygen that helps destroy the anaerobic bacteria. They cannot live in oxygen, so when you put that oxygen down in there from the hydrogen peroxide, it destroys the bacteria. Free-floating bacteria are called star. Those are called planktonic bacteria. Plankton, down in SpongeBob's deep sea, pineapple in the sea, but plankton, planktonic, okay? Down in the sea. He's free floating. He doesn't really have any friends. He kind of tries to just find his own way in life, I guess, by doing bad things, but it doesn't really go good for him, okay? Anyways, planktonic. It's only about 1% of all bacteria. Like I said, it's fittest of the survival. Did I say that right? Fittest of the... Well, anyways, it's... These bacteria know they need to attach to something, otherwise they're going to be free floating and they're more susceptible to be swept away and killed, okay? They need to attach to a pellicle, make their matrix, okay? They need to protect themselves, so we'll get deeper into this, but they need to survive. And they know that if they're floating around, they're not going to survive very long, so there's not many of them doing that, okay? So planktonic, there's about 1% of all bacteria. They rarely exist as planktonic because they know that they will die. They've got to get down and attach. More susceptible to antibiotics. They can be easily flushed out of the pocket. They know they're going to die if they're free floating, so they know they need to attach. Okay, so there's not many of them. About 99% of all bacteria is attached. See how this all goes together? So they attach to each other and to surfaces. Once they attach to a surface, a bacterium exhibits characteristics that are different from those that if it had as free-floating bacteria. So they're basically like, put me in, coach. You know, so these bacteria, once they attach to something, they have a job to do. Now they're not just free-floating looking for things. Now they're like, put me in, coach. I want to work. I'm going to do something. So their characteristics change a little bit, okay? So free-floating bacteria, nope, not good. So once they attach, they have a job to do. They want to, you know, contribute to the team. What can I do, coach? They will form and adhere to a structure called biofilm. Well, we know they attach to the pellicle and they create the biofilm and stuff like that, so it's not that bad. Biofilms. It's a complex and dynamic microbial community containing many types of microbial species embedded within a self-productive matrix. So basically what that means is you've got a whole bunch of different kinds of bacteria that all contribute to something else. Somebody has brains, somebody has muscles, somebody has whatever else may be the case, you know. You've got a dentist, you've got a doctor, you've got a hairdresser, you've got an athlete, you've got a construction worker, you know, you've got all these different bacterias that contribute to something different and they work as a team. So they're very complex and dynamic, and they are a community because these are little microorganisms, living things that are trying to get a job done together. They all want to survive, so they're going to work together to do so. Some are more better at it than others, and we'll get more into that too. So these microbes in the biofilm are going to secrete that matrix to protect themselves, okay? Because they know if they don't put that matrix up, they can get brushed away or, you know, flossed away. As long as we dislodge them, they're gone, you know, so they need that matrix. So a matrix is just a slimy barrier of sugars and proteins. That's why they say don't drink sugars and stuff like that, and certain carbohydrates that have proteins in them, blah, blah, blah. We'll get deeper into that too, but they feed off of that stuff. So their matrix is a slimy barrier that consists of sugars and proteins, right? Proteins are amino acids, are building blocks. So anytime you have a protein, that's a building block to something. So sugar helps their metabolism We'll get deeper into that, but sugar and protein. So they need those building blocks and sugars. You know how they say we get like on a sugar high? That's like what these bacteria do. That's how they survive. They can ferment it and gives them power. Their powerhouse. It gives them metabolism. It gives them energy to keep going. And then they have building blocks, the proteins. Okay. And they adhere to living or non-living structures. Okay. Polymicrobial means several different species of organisms are present. So if they're poly, many microbes just means a bunch of different species. Biofilm is found everywhere in nature, not just in oral health. It has a major impact on human health, though. It's everywhere, and it is everywhere. It's good for us, bad for us. It's everywhere. For us, biofilm can be found on medical and dental implants, contact lenses, believe it or not, pacemakers, artificial joints. We know about that. That's why people need a pre-med, because bacteria and biofilm can form there. For us, it's in our dental unit water and suction lines, and then on teeth as well, you know. So biofilm formation, there is a star here, okay? So we know they start as free-floating bacteria, but they form to biofilm quickly. They have an urgency to hurry up and attach. They're floating looking, hey, who needs me? Who needs me? Hurry, put me in, coach. What can I do? What can I do? You know, they're looking for somewhere, okay? And then once they actually attach, they have a job to do now, so their characteristics change, okay? So within minutes, these free-floating microbes attach to the surface, ... if the community allows them. We'll get deeper into that. But it's very selective who they let in. These bacteria, they're smart. They're like, we've already got a dentist. We need a doctor. Or we already have a construction worker. Now we need an interior designer. You know, like they're very selective. So we'll get deeper into that. Okay, so free-floating bacteria forms quickly to the biofilm, okay? Within minutes, those free-floating bacteria attach to a surface, if they can, okay? 2 to 4 hours, microbes produce the protective matrix and become resistant to antiseptics and antibodies. Anytime you hear about this matrix, it's like their own little capsule, like their own little igloo or something that protects them from being mechanically washed off. Actually, I don't know if that's proper, but it helps protect them more to antiseptics, so you can't just Listerine and wash them away. They're actually attached there. They have a matrix, which is a slimy biofilm, so they're attached. You know, it's not really going anywhere easily, I should say. So antibiotics won't penetrate it. Antibiotics and antiseptics, nope, you do need the mechanical brush, okay? That's within 2 to 4 hours. So within minutes, free-floating bacteria attach, okay? 2 to 4 hours, they produce their protective matrix and become resistant. I don't know if this is right, because then it says 6 to 12 hours, microbes produce protective matrix and become resistant to antiseptics and antibiotics. Let me check on that. 2 to 4 days, fully mature. All right, 1 second. So we're going to go perio. We're going to look at his slide really quick. We have oral biofilms here. Let's look at this just to make sure this is my error and not his. Okay, we want to be efficient. But still, free-floating, within minutes, they attach. And let's see, microbial communities, here we are. Okay, so within 2 to 4 hours, I gotcha. Free-floating microbes attach to the surface within minutes. In 2 to 4 hours, microbes form strongly attached microcolonies. So they don't do their matrix yet in 2 to 4 hours, okay? But they're attaching pretty strongly now. Now they're getting stronger. They're building on top of building, okay? And in 6 to 12 hours is when they produce their matrix, okay? So 2 to 4 hours, they're just strongly attaching now. They're attaching, gaining friends. Let's get some more. Let's attach here. Once they have the right ingredients and the right people, bacterias, right, living organisms, then in 6 to 12 hours, they're going to produce that matrix. Now it's getting a little harder to get them off the tooth, okay? That's within 6 to 12 hours, the matrix, okay? 2 to 4 hours, just strongly attached. In 2 to 4 days, they are fully mature biofilm colonies and can rapidly recover from mechanical disruption and reform within 24 hours. Okay, I gotcha. So, free-floating bacteria forms the biofilm quickly, okay, within minutes, the free-floating attached to the surface. 2 to 4 hours, they got more, they are attaching tightly, pretty tightly. 6 to 12 hours, they produce their matrix, okay, becoming resistant to the antiseptics and antibiotics. That stuff's not going to penetrate them now. It's a little rough and tough there. It's their igloo. And then 2 to 4 days, they are fully matured. Once they matured and you disrupt them, they could rapidly reattach within 24 hours, okay? It doesn't take 2 to 4 days. 2 to 4 days is out of the window. Once they're fully matured, it's only going to take them 24 hours, okay? They know what they're doing now. Now they know how to adapt. They know how it works, okay? They're not new to it anymore. So once they're mature, you disrupt them and you don't get them all the way out, they're going to attach again within 24 hours, okay? I hope that helped. We're almost halfway done already. So far, biofilm formation, they colonize to the tooth surface. Microbes join with early colonies, so there's early infection. And then 3, complex microbial community develops and chronic infection. There's no star there. That was just one of his PowerPoints. So biofilm protects itself from the host immune system, antimicrobial, and environmental stresses. No star. Here's a star. What are biofilm's 3 features that enable microbes to tolerate external stresses? Meaning, what does it use to protect itself? What does biofilm use to protect itself? Here's a star. 3. So 1 is blocking. 2 is mutual protection. 3 is hibernation, okay? So what is it going to do? What does bacteria and their biofilm do to protect itself? Blocking. So the protective matrix, which is their igloo, prevents large molecules, meaning antibodies and inflammatory cells, from penetrating the biofilm matrix. So their igloo, so antibodies, large molecules, stuff like that is not going to penetrate their igloo. They're going to block it, okay? They've got their matrix, they are protected, okay? Mutual protection, meaning the different species of bacteria offer protection effects to other bacteria, okay? So existing microbes in the normal flora of the mouth may offer protection to the host. So there's 2 different ways. So mutual protection. So all these different microorganisms, all these different bacteria in their colony. Remember, we've got the carpenter, we've got the construction worker, the hairdresser, the bodybuilder, the leg lifter. All these people are going to contribute to something different and they can have each other's back. Mutual protection. They all have something to offer and they'll protect each other because it's survival of the fittest. Not fittest of the survival of the fittest. Everyone knows what they need to do to protect themselves and they can't do it alone. They need to work together. So they're going to mutually protect each other, and then our own microbes will help us and our immune system protect us, right? We like our good bacteria. And then hibernation. So bacteria hibernate and are unaffected by antibiotics. So one way that they can protect themselves is hibernate. I would like to know more about that, but that's not going to be on the test. Okay, extracellular slime layer. This sounds like a matrix, doesn't it? So star, the protective factor that can prevent biofilm from being killed with antibiotics, antimicrobials, or the body's immune system. Well, we know that if it's an extracellular slime layer, that's sounding like their matrix. So it's just another fancy name for their matrix. And when they have that igloo, no antibiotics is going to penetrate that because it's larger molecules. No antimicrobials are just going to rinse it away because it's attached. It's their igloo now. And your body's immune system is not going to do that either. Commensal bacteria, that's just bacteria. It refers to a type of relationship between two or more different organisms that eat from the same fish or the same dish. Oh my gosh. So basically it's a win-win. It's a neutral relationship is all that is. All epithelial lined surfaces are colonized by commensal bacteria. Everyone's harmony. Symbiosis, living in harmony. Everyone's symbiosis. Everyone's living in harmony between us and our commensal organisms. Nobody's a threat. We're all just getting along. Dysbiosis, living in disharmony. So it's a microbial imbalance on or in the body. Certain bacteria become dominant. I want to say it's cocci and rods. I could be wrong, but I think it is. It's not on here, so I'm sure it'll be on another one. Initial dysbiosis can initiate gingivitis. Obviously, if there's an imbalance, there's going to get some disease started. It's established over an extended period of time. It can trigger an inappropriate and excessive host response, resulting in periodontal tissue damage. Okay, we know that. Dysbiosis, disharmony, there's a balance tip. Stuff's going to go wrong. Control of microbial growth. Star. Control of microbial growth. Let's see. So, this says bacteria living in biofilm are more resistant to antibiotics than free-floating bacteria. We know this because the biofilm is going to produce its matrix to protect itself from larger molecules like antibiotics. To kill bacteria in biofilm, they need 1,500 times the amount of antibiotics as free-floating bacteria, which is lethal to patients. So that doesn't really matter. But what does is antimicrobials work best in tandem with mechanical disruption. We know this. So Listerine is only going to be good if you're using brushing and flossing as well. So you want to floss to dislodge. You want to brush to dislodge plaque and then rinse to kill planktonic bacteria. How is it best achieved to control bacteria in dental plaque biofilms? Okay, there's a star here. How is it best achieved to control bacteria in dental? Oh gosh. Control of bacteria in dental plaque is best achieved by physical disruption of the plaque by brushing, flossing, and periodontal instrumentation. We know that. How can plaque be transmitted? It can be transmitted, bacteria can be transmitted from the oral cavity of one person to another, typically kissing. Okay, here we go. Star. 5 stages of polymicrobial biofilm formation. 5 stages of different kinds of bacteria in this biofilm formation. So how is it all going to form? You have 5 stages. So stage 1 is the initial attachment of microbes to the pellicle. We know this. So they're free floating and then they're going to attach, right? And then 2, they're going to permanently attach. Within 2 to 4 hours, they're going to get that stronger connection, that stronger build on build on build. Stage 3 is maturation stage 1. It's the self-protective matrix formation. So we know those 3 steps. So stage 1, 2, and 3. So stage 1, initial attachment. Stage 2, permanent attachment, more stronger. They don't have the matrix, but they're attaching pretty strong. Stage 3, protective matrix. Okay, maturation, protective matrix. Stage 4, maturation stage 2, which only simply means now they're starting to get a mushroom-shaped microcolony. So they're going to kind of grow out in a dome. So instead of just being an igloo, now they're going to be a little pedunculated with a little stalk and a mushroom cap, okay? Stage 5 is dispersion, escape from the matrix. So basically what they want to do is they want to grow. Survival of the fittest, you take up space, you can only do so much, and when it's time to move on, you do. You move on to somewhere that's even better, has more space, more minerals, things to eat. That's what these bacteria do. So once they're so big, they're matured, they're like, I know what I'm doing. If you dislodge me, I'm going to come right back in 24 hours, okay? And that happens within what, 2 to 4 days? I don't know. But anyways, so yep, so we'll know 6 to 12 hours is that matrix, and then 2 to 4 days is the complete maturation. So they are matured. They're getting out in their mushroom shape because they're growing, growing, growing. They need more space, and then they're going to disperse to different areas, okay? So stage 1, star. Initial attachment of microbes to the pellicle. Within minutes after cleaning, a film forms over the tooth, the acquired pellicle. We know this. Composed of salivary glycoproteins and antibodies. The purpose of the pellicle is to protect the enamel from acidic activity. So we do want a pellicle on there. We just don't want all the bacteria to attach to it. Within hours, free-floating microbes begin to attach to the pellicle. Well, whatever. We know that it can take minutes. Those free-floating bacteria can attach within minutes. We know 2 to 4 hours are just going to form a little bit stronger, right? So, and then 6 to 12 hours, they're going to form their matrix, and 2 to 4 days, they're going to be fully matured. Okay, so we have stage 2. Okay, stage 2, star, permanent attachment. This is within your 6 to 12 hours. I don't know if that's what this says, but yep, 6 to 12 hours. That better attachment, permanent attachment. Microbes are able to withstand hydrodynamic forces, which just means fluid, and obtain permanent attachment. So that permanent attachment, no antiseptic is going to wash them away. Attached microbes produce substances that attract other microbes to the community. So quorum sensing does not happen until stage 4, okay? So in stage 3, what they're doing is, this is stage 2. So in stage 2, they're permanently attached, and they're growing, they're permanently attached, and they want more. They need more people. They need a mathematician. They need somebody else to help complete this matrix, okay? So they don't have a matrix. They need to pull some people in. So what they're going to do is they're going to produce substances that attract other microbes to the community. So they're not really, I wouldn't say they're really communicating yet, but they are releasing something to attract them over here. So you're gonna be like, put out something for a mathematician, like, hey guys, we got some stuff going on. Who wants to be a part of this? Okay. It's called, co-aggregation is when genetically distinct bacteria become attached to one another. Early colonizers determine which microbes will colonize later. So if you get your cockeyed rods, they're like, I think we need some spirochetes or spirochetes and some fimbriae or whatever they're called. They're going to try to attract those types of bacteria. But it's not quorum sensing until stage 4, okay? I don't know why, but it is. So stage 3 is your maturation stage, okay? It's your stage 1 maturation. So it's self-productive matrix. So stage 3 is their matrix. They are firmly attached bacteria, secrete surrounding protective substance called extracellular protective matrix. We know this usually consists of proteins, glycoproteins, and bacterial DNA. So glycoproteins, I believe, are just like sugar proteins, like sugar building blocks, because they need proteins and sugar. So you have proteins, which are your building blocks. You have glycoproteins, which are sugar building blocks, and then bacterial DNA. So obviously you're going to have the bacterial DNA in their matrix because they're putting their own little oomph into it. Microbes become cocooned in the protective matrix. We know this. Protects against host-generated immune systems. So in this stage, in stage 3, when they have their matrix, this is when chronic disease is established. This is periodontitis, okay? Stage 4, here we go. This is when we have, this is when really the quorum sensing is indicated, okay? So maturation stage 2. In stage 4, they have their mushroom cap, right? And their peduncle, they need to grow outward because they need some more space because they keep gaining these people, well, these living organisms coming in, they got all these people to help. Let's protect each other. We need to grow. This is, we need to stay protected. They cluster together, form a mushroom shape, attached by a narrow base. They're pedunculated. The microbial blooms develop when specific species grow at an accelerated rate. So, wow, these species are growing. Certain species, they're accelerating. They need some more space. Biofilm becomes thicker, stacking one species on top of another. You know, they're polymicrobial. Fluid channels penetrate the protective matrix, but those fluid channels are not antiseptic. So the fluid channels are what bring nutrients and oxygen to the microbes and carry the waste away. So they're growing so big, they have this big mushroom cap in stage 4. There's so many different kinds of different kinds of bacteria. There's so many of them growing at an accelerated rate. They need space. They're growing up and out. Then they have to be nutrient, right? So they're losing space. They're losing nutrients because there's so many of them. So they have these water channels come through that feed them and give them the nutrients they need and oxygen, because these are not subgingival. These are supergingival. So they're going to get oxygen. We breathe in, they're all going to get it. They have to be aerobic, right? If they're sub-G, they're anaerobic because they don't like the oxygen. Fun fact. So the fluids, the fluid channels that bring the nutrients and oxygens and stuff, those include saliva and beverages that we consume. So if you're drinking sugary stuff, they love it. They're going to use that for their extracellular matrix because they love their glycoproteins to help build with, right? So direct cell-to-cell communication occurs in this stage via chemical signals resulting in transfer of genes among microbes. So quorum sensing. So what they're doing now is they're transferring parts of themselves. They're transferring genes, okay, for quorum sensing. And I've got stuff on this. You ready? He is going to ask about quorum sensing. So what is quorum sensing? It's when the microbes in the biofilm release and sense small proteins to communicate with each other. So they're releasing parts of proteins of themselves, you know, they're releasing genes of themselves, these proteins of themselves, their own building blocks, you could say, to communicate with each other. It is used to trigger events like adhesion of additional microbes and formation of protective matrix. So basically, they're signaling people now intentionally. They're like, all right, I'm going to communicate with you. I'm sending that. Here's part of my gene, and you're going to fit here, and you're going to like it. This is where you need to be. This is where the party is at. It allows bacteria to communicate and coordinate their behavior for growth. So now they're quorum sensing. Now they're all communicating. Now they're all mature. They're in their mushroom cap. They know what's going on. Okay. And they're surviving. It occurs in interspecies too. So it doesn't matter what kind of species they are. They can communicate to each other now, and thrive. You know, now they're like full force. They are mature. They are communicating in stage 4. Okay. Stage 5, there's a star here. Dispersion. This is the escape from the matrix. So basically, microbes disperse from the colony to spread and colonize to other tooth surfaces. Survival of the fittest. There's not enough nutrients, not enough space. They're going to go and grow into a new area. Like, I live in Mooresville, it's getting overcrowded, so I want to go move to a new area, you know, where there's more land and I have more space to myself. Okay, that's what they're going to do. Okay. So, bacteria attach subgingivally to the tooth surface, epithelial lining, other bacteria already attached to the tooth or epithelial lining. Some bacteria may remain free floating in the sulcus because remember, sulcus is a fluid-filled space, so they can just kind of hang out in the sulcus, but they can be washed away pretty easily. Okay, no star there. Bacteria attached to the tooth. Cockaian rods dominate tooth-associated plaque biofilms. So it is the cockaian rods that get this party started, okay? They are the ones that dominate this plaque biofilm, okay? Cockaian rods. Once you get more into periodontal stuff going on, then you're going to get some periodontal, bacterias. We'll get into that too. But this is just like your Streptococcus mutans or whatever, all that good stuff. Okay, he's not going to ask names about that. Bacteria attached to the epithelium. So, distinctly different from bacteria that attach to the tooth. Okay, so these are bacteria that attaches to the epithelium. So they're going to attach to our gums. So these are the ones that are going to want to get periodontally involved because they're already there down in the gums. They're on the gums, in the gums. So these contain large number of spirochetes and flagellated bacteria and gram-negative cocci rods. Okay, okay. So basically, these are more periodontally involved. If they're gram-negative, they're anaerobic. They don't like oxygen. So these spirochetes and flagellated, which just means they're a little bacteria with a little tail, and then the windy noodle looking ones. So those ones, and then gram-negative cocci and rods, they're going to go down under. Okay, they are epithelial attached. They can invade gingival connective tissue and be found within the connective tissues on the surface of the alveolar bone. Those are the ones that are going to make your immune response resorb the bone, okay? Spirochetes, they're spelled like spirochetes, like, I don't know, spirochetes and flagellated bacteria. And then gram-negative cocci rods, okay? Almost done. Hypothesis on role of oral biofilms in dental disease. So, there's 5 of them. We only have to really know 2. So, hypothesis on role of oral biofilms in dental disease. So, this is just a hypothesis. It's an educated guess, okay? So, 1 and 2, there's specific plaque, 1, non-specific plaque hypothesis, 2, specific plaque hypothesis. These are the 2 we need to know. 3 is etiological plaque hypothesis. 4, microbial homeostasis with host response, and keystone pathogen. We'll get into those later, but 1 and 2 we're going to focus on because that's what's bolded. Non-specific plaque hypothesis and specific plaque hypothesis. And what that means is, so, boom. 2 hypotheses that focus on the role of numbers of bacteria and specific bacteria that present in oral biofilms. So that means these are the 2 hypotheses that want to deal with the roles of the numbers and the specific, so how many bacteria and what kinds of bacteria. That's what these 2 are more involved in. So non-specific plaque hypothesis is just an educated guess stating that the amount of bacteria in the biofilm adjacent to the gingival margin leads to gingival inflammation and subsequent periodontal destruction. Basically, all plaque has the same bacteria. We know that's false. But anyway, so this is basically saying, so non-specific means that all plaque has the same bacteria. Problems with non-specific is that it fails to explain why most cases of gingivitis do not progress to periodontitis. If we had the same biofilm, if we all had the same bacteria in our biofilm, how come some of us have gingivitis that never go into periodontitis? Why does it never progress? Or, vice versa. So that's why. It's thought to be too simplistic. Specific plaque / microbial shift hypothesis. There's a star here. We need to know this one. So basically, this is saying microbial composition of the oral biofilm rather than the amount is the deciding factor. So it depends on the type of bacteria. So an increase in the number of specific pathogens is associated with periodontitis. That's what this is saying. So what's highlighted is bacteria in the biofilm change from predominantly gram-positive aerobic community to one consisting of groups more of a gram-negative. So basically, for periodontitis, this is an educated guess. There's not enough research. That is first gram-positive, and then you're going to have more of the gram-negatives, which causes the periodontal disease. Okay. This is a star. We need to know this. We are almost done. So Kransky's microbial complexes, there's a star here. So this is with the different colors and the arrangements of different bacteria is what they do. So basically, so Kransky's, if I could say it, S-O-C-R-A-N-K-Y, apostrophe S. So Kransky's It's grouped microorganisms into complexes and assigned each a complex color. So blue does this, purple does this, orange, red. Okay. Orange and red are thought to be the major agents in periodontal disease. So remember gram negative, right? Gram negative stains red is bad. Those are periodontal involved pathogens, right? So orange and red, periodontal disease, okay? Yellow, green, blue, and purple. Yellow, green, blue, and purple were thought to be compatible with gingival health. Well, yellow, green, and blue, okay, those are primary colors, aren't they? Something like that. Maybe not quite. I think red's one of them. But anyway, so yellow, green, and blue. I can't think of anything that's yellow, green, and blue, except for a pretty butterfly. Yellow, green, and blue, and purple. We know purple's us. We know that has to do with good gingival health, right? And the gram-positive staining. So yellow, green, and blue, okay, are great colors. They're everything except for orange and red, really. No, orange and red is bad. That's periodontal. Boom, we don't like that. So yellow, green, blue, and purple, us, are thought to be compatible with gingival health, so good health, okay? Problems with specific plaque. So problems with this theory is that, okay, so red complex organisms can be found in the absence of periodontal disease. So are they truly pathogens? So these ones that are red and orange, they're there without periodontal disease. So are those the cause of periodontal disease? That's a problem. Mainly new recognized organisms have a better association with periodontal disease than those categorized in the red complex. So basically there's some new bacteria that they're finding that can really initiate periodontal disease that are not even in the red. So that makes it bad. Contemporary perspectives on role of bacteria. I don't think we need to know this. A pathogenic microbial biofilm is a prerequisite, but the presence of pathogenic. Okay, so basically what this is saying is there's no current evidence to support the argument that red complex bacteria are potent indicators, initiators of disease. There's no star on that. We don't have to worry about that. Okay. I don't see any other stars on these. I just included these. Oh, I do see one more star. Okay. Okay. So one more star. Contemporary perspectives on role of bacteria. So contemporary perspectives. The one we need to know is microbial homeostasis is a host response. Okay, so microbial homeostasis, well, we know that there's a microbe balance, right, with the host response. So microbial homeostasis, host response hypothesis. So plaque biofilms are the cause of initial inflammatory response leading to gingivitis. Pathogenic bacteria are not the direct cause of periodontal tissue destruction. Its host-related factors are the major factors that contribute to initiation and progression of periodontal disease. Page and Schroeder established that gingivitis does not progress to periodontitis unless another unknown factor tips the biofilm balance further to destruction. So this is really just that balance we were talking about in chapter 12. What do we believe? We believe clinically healthy periodontal tissue maintains a highly ordered, mild state of subclinical inflammation. I don't know if that makes sense. So basically what we believe is the shift. There's a balance. There's got to be some kind of shift. Bad bacteria taking over, so it's going to initiate all this stuff going on, and then our host inflammatory response contributes to the tissue destruction. Woo! And that is all of chapter 13. We have completed all of our chapters. We're going to go kick butt tomorrow on this exam. I hope this helped. I know I'm done for the night. I'm going to watch these over maybe once, and then I'm going to go to bed. But I don't know. We got to do theory too, so we'll see. But I hope that helped. Thank you. Bye.